So I was looking back through my slides and I saw that I started talking about a couple different cohorts, but it's actually been growing. So I'm going to give you an update there. I wanted to start by addressing, a lot of people have talked about this movie and how it could be somewhat representative of the exposome. But it is a bit reminiscent to me of this totality issue that let a lot of people be dismissive of the exposome because it was an impossibility. So I'm looking forward to the sequel, which is the most important things at the most important locations at the critical time. So this will be the affordable sequel to that movie. That's what I think we're going to focus on today. So we've talked much about this. You know, Chris Wilde started this whole activity. My group's been thinking a lot about this for many years. And as has been pointed out, you know, when we talk about what the exposome is, we are thinking about the physical, chemical, ecosystems, lifestyle, social, all of these things need to be part of it. But I think a really key message is that for any of these external forces to have an effect on our health, they have to alter our biology. And so when we think about things that maybe happened 20 or 30 years ago, if you had an exposure that is increasing your incidence of a disease, there has to be a biological underpinning to that. So whether it be an epigenetic change, protein modification, a change in a system. And so the idea is that by looking at those human outcomes and processes, we can figure a lot of that out. I like to think about if I go to the doctor today, I am the summation of whatever's happened over the past decades of my life. The doctor, even if we get information, we can go back and kind of back calculate what exposures were there, but you're still just who you are when you walk in. They can take blood out of you, they can do a bunch of tests, and the idea is that if you are unhealthy because of your past life, there's markers of that. And we should really think about using the biology to let us think about what's happened in the past. So this has been a great figure as I've been putting it together over the years. It started with, you know, we have like over a century of the human genome, but exposomics is still a relatively young field. But it's really growing rapidly. If you look at the number of papers that cite exposome, and all the different conferences and things, a lot of the stuff that that our hosts here have done across multiple symposium now all throughout the world. As noticed, we now have a journal in the field, and really have a lot of exciting progress coming through here. As was mentioned, we held this conference at Cold Spring Harbor Laboratories back in December. And I think this is a really important effort in that Cold Spring Harbor Laboratories has been known for really being instrumental in helping establish multiple fields, a lot on the genetic side. And the idea of the Banbury conferences is to essentially lock a bunch of people in a room for 3 days, make them eat all their meals together, and just have them say, we're going to solve problem X, whatever it is. And so I was talking to colleagues at Columbia, Tom Maniotis, anyone who's ever done any cloning. He wrote the manual for that as a Cold Spring Harbor course. And he suggested that a Banbury conference could be very helpful for where exposomics was. Because one of the challenges is that what do we mean by it? What is the definition of the exposome as it's evolved since Chris Wilde's first part of it is? And so, as Rick talked about yesterday, this was the definition that the group came up with. And it was 23 people from, one of the rules was that we could only have one person from each institution, so it was like very much distributed across primarily the United States. We had a few Europeans on it, and from very different disciplines. People that did, ontologies and data science and geospatial work, physicians, molecular biologists, toxicologists, a very broad group. But the idea was really to think about, what we mean by it, and then how you operationalize it. And so the idea is that we're looking at these comprehensive and cumulative effects, physical, chemical, biological, and psychosocial. But the goal is really to enable discovery-based analysis of the environmental influences on health. So we published a meeting report. The meeting report really talks more about the process and not the results. The results and the definition are part of a manuscript, which I've distributed now to the participants. We're hoping to submit it in about a week and a half. but we work closely with editors at a high-profile journal, so we're hopeful that it'll get in and out to a very good place that should have very wide distribution. But the goal here is really how, in order to integrate exposomics into the entire biomedical enterprise, we have to have very consistent and actionable definitions. And that was a big part of this initiative, was to come up with this way that we could work on that. I do know that our colleagues in Europe that are part of that IHIN network that was mentioned, they are planning a meeting this fall to kind of build upon this and think about the next steps in the strategy. Essentially, they're pretty much adopting this definition. Some of those key leaders are at the meeting. And following up on our sort of genetic thing, the plan is to have it at the Mendel Museum, where Gregor Mendel did his work, pioneering work on genetics. So I think we're really making strides here and kind of formulating how we want to do things. So kind of building off of what we heard from Craig the other day is, so I think all of these factors are very important for exposomics. But as a scientist, I need to do what I can do. I can't do everything. So my lab focuses on using mass spectrometry to measure as much as we can in biological samples. Primarily focused on plasma, but we've done things on cerebral spinal fluid, tissue, saliva, tears. We can do it in all sorts of different things, but we focused on plasma because biobank samples are readily available. And when we looked at possible ways of using things, that was the place where I thought we could have the biggest effect. And so what we do is a combination of liquid chromatography and gas chromatography, high resolution mass spectrometry. There's a lot of data processing and things go on with this, and network and pathway analysis. But we've worked on this workflow very carefully to get as much coverage, as deep coverage as we can, but always thinking about the cost of this. And so the idea is you're looking at these small molecules in a sample. You're picking up endogenous molecules or normal biochemistry things, and all these exogenous compounds. And so the GC Orbitrap, which is a relatively newer instrument, is very good at detecting these more environmental chemicals. And so by using both of these approaches, we get very good coverage of chemical space. We can detect hundreds of thousands of peaks with varying levels of confidence in what they actually are. And so one of the challenges is how we look at what these things are, is how we identify it. And a lot of this has come from how the field of metabolomics has viewed this. So Emma Szymanski worked with a team to develop this Szymanski scale about how we identify those molecules. And the goal is always to get this sort of level 1, level 2, where you have a reference standard and have very good accurate matches. And I like to compare this to that vehicle identification that you have in an automobile. That little number there you see typed out, and you'll see it on your registration, which no other vehicle in the world has that VIN. Like, it's specific to that vehicle. But if someone drove by your yard and stole your puppy and drove away with it, and the police came and said, did you get the VIN number of that car? You would say, no, I couldn't see the VIN, but I got part of the license plate and I saw it was a blue Toyota. You'd have enough information for the police to track down that vehicle. You don't always have to have that perfection to be able to get that identification, especially if you have large cohorts and large effects where you're seeing these repeated effects. But the idea is we do want to get there ultimately, but there's a lot we can do with less than perfect measurement. And so the challenge here is that in metabolomics and using some of the different systems, it doesn't always transfer over to other approaches like with this gas chromatography-based system. And so several of us led by Crystal Pollitt and Doug Walker, we formed a GC Orbitrap users group that started meeting a few years ago, and we came up with a scoring framework that was kind of parallel to the Szymanski scale, but worked better for the GC type of work we were doing, because it just didn't transfer some of the retention indices and things, which I won't really get into. And so it's been a lot like, how do we use these systems to be more appropriate for exposomics? That's always been the goal, to make it more suitable for purpose there. And then we think how to apply that. So we have this paper, this came out a couple weeks ago in Nature Neuroscience, looking at how we can use that high resolution mass spectrometry to characterize the chemical aspects of the exposome for neurodegeneration, which obviously is very apropos for our meeting here. And it's a complicated process. So this is a paper that was just accepted on Friday in ES&T. My postdoc in July really did a heroic effort to get people from 20 different institutions and 7 different countries, over 400 references here, to really look at how you can use high-resolution mass spectrometry for exposomics. And again, thinking that we have this goal of trying to really figure out the environment part of it. So it's been great, like trying to get people together to think about these things and think about the tools we're using for exposomics to really drive the field forward. So hopefully that'll come out in a couple weeks. I don't know exactly what their publication turnaround is. But it's been very important to get people to understand that there are nuances with exposomics, and that if we put our effort towards it, we can really improve how we're doing things. So there's been some mention about drugs and different xenobiotics, and I want to point out an approach that we've had taken over the past several years and are still moving forward with it, is when I talk to clinicians, I like to give this example of grapefruit juice. I think Bob mentioned this yesterday too. The furocoumarins in it inhibits cytochrome P450-3A4, and it really disrupts your metabolism of many common drugs. And I like to just pose the question is that if you think chemicals in grapefruit juice are the only chemicals that alter P450s and other enzymes, you're crazy. I mean, there's thousands of things that do this. But we don't think about how the environment influences how the drugs we take work. And one of the challenges in looking at these things is that we have reference standards for most common drugs, but not for all their metabolites. And so as part of a Common Fund project that Dean Jones led, we worked on a project where we took 96 well trays and put liver fractions into that tray. And we would put a defined chemical into that well. And then we would take samples every few hours in that time. And so what you end up seeing is metabolites of that parent drug get formed during that time. You know where they came from because you're the one that put the chemical in there. But you could see, and because you're using liver fraction, you get phase 1 and phase 2. But we would then put those identities into the databases to say, so it's not really a standard, it's more of a pseudo-standard, but you know that that metabolite had to have come from that parent drug. And so when we went through this process, Ken Liu, the postdoc, said, he's like, I think I found a novel metabolite of caffeine. And it's like, oh, come on, people have been studying caffeine for decades. And so we went back and did a stable isotope labeling of it, took it through, and sure enough, this was a new unreported metabolite of caffeine. And so the idea is that when you're doing this in an unbiased, untargeted way, you can just find things that people didn't know existed, but now they're there for future identification. And so the idea was to do this for many different drugs and environmental chemicals. You could then inject a mouse with that parent compound, and we would see the metabolites that we identified in the in vitro system. We could even go into human cases and find the different metabolites of various drugs in that same way. So really kind of trying to build up our ability to identify the metabolites of these various xenobiotics that we think are important for human health. And so again, we spent a lot of time developing a lot of this methodology, but ultimately what we wanted to do was study biology. So this was one of the first projects we did looking at Alzheimer's disease, started probably in 2017. And this was about 200 total people looking at Alzheimer's disease, mild cognitive impairment. And we had plasma samples from them. And we went through and did our kind of metabolome-wide association study and found things like glutamine, which people had reported before as being elevated, these piperine compounds, which are pepper derivatives. We don't think it's really involved in it, but when people develop diseases, they change their habits, change how they eat and things, and so probably less pepper consumption from that. But we found this metabolite of DDE, and I recognized that a colleague of my former postdoc had published a paper a couple years earlier where he looked at pesticides in Alzheimer's disease and had seen elevations in DDE metabolites. And so we had this sort of orthogonal confirmation even before we came out with this, that this is one of the challenges too when looking at environmental chemicals. So if you look at this paper that was published in 2019, you will not see that result because the reviewers said you need to have a higher cutoff where if you don't see that compound in like the majority of the patients, you can't include it. But if you're looking at a disease caused by an environmental chemical, it's usually only in a subset. But at that time, like, we couldn't convince the reviewers that we should include it, even though we believed it and it was there and there was other reasons to look at it. But it was like, well, we'll pick our battles and went on with that. But it shows one of these challenges when you're looking at exposomics. You don't always want to be judged by the way that people think of metabolomics. Like, think if you did not see components of the Krebs cycle in people, that would be a problem. And so it's understandable why you would want to say, yeah, we see it in the majority of patients. But if you're looking at someone that has a chemical exposure, it might only be 10% of the population that has it, but that 10% might have an increased risk of a certain disease. And so again, just how we kind of approach these things has to change. We also detected adducts of rivastigmine, which is one of the drugs that's used to treat Alzheimer's disease. And this was also kind of a funny thing, like as I look back at this years later, like we didn't include rivastigmine in the results because we kind of thought it was an artifact. Like the idea of the drugs that people take, like of course we detect those because they're taking the drug. And not really thinking of like, well wait, it's an important piece of information because what are the levels of those metabolites and how do you detect those things? And I'll come back to how we've been trying to address that. So about 6 years ago I moved to Columbia. I was very interested in continuing this work on Alzheimer's disease. And I was introduced to Richard Mayu. He leads the YCAP study, which is the Washington Heights and Inwood Community Aging Project. Now has over 5,000 people in the study. And the catchment area is about 50% Hispanic, 25% Black, and 25% white. So he'd been studying this population for decades, had done all sorts of genomics, all sorts of phenotypic analysis on the clinical side, and he collected blood. So he had plasma from all these individuals. And so I said, hey, can I have some of that? And so we wrote a grant and started working on that. He had seen an increased incidence in the Hispanic population in YCAP, and most of them are from Dominican Republic descent. So he launched the AFIGA study, which is based in the DR, and now we've enrolled well over 1,000 people, actually have spinal taps and CSF from about 25% of them. And then Jennifer Manley at Columbia was working closely with Richard, and she was, as the people in YCAP aged, she's like, well, you know, their children now are adults and they're getting older. We should study the offspring of the original YCAP. So she started the YCAP offspring cohort, and so we've been talking to her about it. And then more recently, Jose Lusinger, he had been looking at the Diabetes Prevention Program Outcome Study. This was a study looking at, I think, metformin. This was many years ago, looking at diabetes prevention. And because people with diabetes have an increased risk of Alzheimer's, he went back decades later and did a full analysis of Alzheimer's disease, cognitive function and things, brain imaging. And so this seemed like another place where we could look at this. And then also this RAND study that Yakov Stern and Chris Hayback run is a study of cognitive aging, and I'll talk a bit about that. So one of the first studies we did with YCAP was just a relatively small pilot study, 160 people, equally divided, cases, controls, and a third of each of the racial ethnic groups. And so we could easily separate out cases from controls by the mass spec. And we also separated people by their race and ethnicity. And I remember Richard showing this data to him, and he's like, that's impossible. He's like, you know, these people are genetically no different. Everything we've ever done, we never see them separate by the racial background. And my first answer was like, well, it could be something as simple as diet. And he's like, well, we did dietary inventories, we didn't see any differences. And I said, diet is not biochemistry. Like, we're measuring these molecules in the people. And so this was very intriguing because if you know that there's different vulnerabilities in those populations and you do a pathway analysis in the different groups, you see that different pathways are altered in these populations that have differential vulnerability. And so this really told us that, you know, this sort of approach can identify these pathways that are altered in these populations that can help us get to this idea, like why we see these disparities in outcomes. So we published this initial report, and I'll come back to what we've been doing with it since then. And so then this AFIGA study, this actually moved along a little bit faster, I think just because the N was a little bit more manageable for us. But we went through and did this initial study in that AFIGA population in the Dominican Republic. And we found some interesting results, but it wasn't terribly compelling. And at the time, some of these new Alzheimer's protein biomarkers, we've heard some about, the p-tau181 and others, were getting a bit more measurable. And so Richard had gone back and working with a group at Columbia, had gone back and measured these protein biomarkers in all of the cases we had looked at, in all controls and disease. And what we found was that if you took the diagnosis of Alzheimer's disease from neurologists who really know what they're doing, and then you look at these biomarkers, it seemed about like 20% of the people that the neurologist said had Alzheimer's disease didn't have the elevations in p-tau. And so if you then said, let's look at the people that have the diagnosis and above the p-tau cutoff, then the data just totally changed. And I think this is something we've seen in so many different conditions. When you're looking at diseases that are very heterogeneous, you're studying different levels of biology, and like you'll never get to it because it's an apples and orange sort of comparison. I think this is a big issue with like an autism too, is you have many different subtypes of the disease. But if you go through and say, let's look at this biomarker positive and clinical diagnosis, is the pathways you see change are totally different. And it shows by having some of these people that they clearly have dementia, but they don't have that AD pathology-based dementia. And so the idea is it showed it was really important to go through and do this in a way where you could see how these certain biomarkers, and what I think is really critical with these biomarkers is they're done on the exact same plasma sample that we're doing all of our analysis on. So we have this very tight concordance across these different markers. And so what we can also do is we can do our kind of metabolome-wide association studies on each of those types of markers. So we can look at the neurofilament light chain, GFAP, and the p-tau181, and we can see that we get hits that correlate to those levels. Essentially, all of the things that correlate to the p-tau181 are also potential biomarkers of the disease, right? Because they're pathways that are changing that same way. And so if you start looking at, it's a bit complicated here, but it's showing things like if you have clinical diagnosis and you're biomarker positive, or you're clinical and biomarker negative, and you go through all these different things, your results are just very different as you break these different levels apart. And so it just shows it's really important to have a very good characterization of the disease process at a biochemical level. And then from this, like we've identified many, high-level identities, like level 1 sort of identities here for looking at things that in the people that are biomarker p-tau positive. These lyso-PCs, I think, are really important. They've been implicated in other studies. Again, we're not looking under the street lamp. We're looking at every sort of peak that comes out. But when we saw these lyso-PCs, it was very interesting because others had reported that. So when we dug deeper into the lyso-PCs, like these are like the different chain lengths that they have of them, there are just so many of these molecules that are being perturbed in this. But we went through and did this analysis looking at this comparison between those plasma levels and for biological AD. And so what's important is these things are involved in a lot of the fatty acid sort of processes and things. And so it makes sense tying into the inflammatory processes. These things are all being disrupted in this way. And so it's really thinking about these things more at a class than as an individual marker. You're seeing how is a family of these lysophosphatidylcholines being altered. And so then we can look at this in a network analysis and look at how, like that p-tau marker, how those things correlate to the other biological markers we have and the diagnosis of the disease. And so like using these ways to tie all this together from these different small molecules. What I think is really important about the network analysis is that you can bring proteins and RNA into this. Like the idea in this sort of multi-omic analysis is, I think this is how we're going to be seeing that it's not always about that individual molecule that's changed, it's pathways and networks that are changed, and we have to really look at it in that way to figure those things out. So I haven't talked too much about environmental chemicals yet, and I'll explain where we are on some of that. But one of the things that was really interesting about this, and we talked about the geocoding things the other day, is my student at the time, Brenda Colley, and now postdoc, said, we have all this data on air pollution here in Manhattan. It's been studied for years by several investigators. We have, all the NO2, the PM10, black carbon, all those things. And so she went through and did these, environmental-wide association studies sort of things, and she found that PM2.5 levels corresponded to alterations in this glutathione intermediate cysteinoglycine. And this, to think about this is like historical exposure, as we've been talking about. If you know where people live and work over time, that you can predict what their levels of exposure are. And then we find things involved in redox homeostasis altered in the people at higher levels of PM2.5. So really connecting that external environment to these internal biological changes that are occurring. And what I'm really excited on this geospatial part, because we have all this detailed history on them, is that it's not just air pollution, right? We've talked about this. We have a lot of data on a lot of external factors from publicly available maps on green space, blue space, built environment, UV radiation. All of these things are in there. And so the idea is if you have people's residential history, you can go through and compile these things, as has been mentioned. But I think what's really critical is then you can tie it back to the biology. I mentioned briefly this study of Yakov Stern. So this was one where, in this RAND study, this is healthy people that were tested for cognitive function under a functional MRI, and they're brought in every 5 years. And he collected blood. That's why, can I have blood? And so we went through and did targeted GC, Orbitrap, untargeted GC and LC, and there's many different chemicals that are elevated and looking at various changes, like to functional or structural changes in the brain. And so we're in the process of analyzing all this, but when you're looking at things like white matter density and different things in different regions, you can connect these exposures of what we see in the plasma that we still measure. Many of them are very persistent chemicals. But I think it's very promising that we're seeing things go in this direction. And so where we are now with YCAP, I've run, my lab has, we've had 600 people that had 3 serial samples. We've run LC and GC on all of those, about 3,600 samples, and we're doing the bioinformatics. And so hopefully in a couple of months I'll have all of this data. For AFIGA, all I was shown was the LC-based work. The GC wasn't done, it is now done, and we're doing the informatics on it. We just started the offspring stuff just last week, and that one with the diabetes project got a really good score, and we're hearing that it's likely to be funded. And in this one, it's over 1,800 individuals, predominantly female, non-white, that have already had MRI and PET stuff done on them, and we'll be doing a combined targeted and untargeted analysis. So really having, what I like about this is that we're not just looking at one population, it's that we have multiple populations to look at and see, are we seeing it in these different groups that have different types of exposures, different backgrounds. But I think it'll really create a very rich data set that'll help us figure some of these things out. As a biologist, I still want to kind of figure out what's going on with this. Just shows that all of this work I've shown on the mass spectrometry works perfectly well in C. elegans, and so we actually use this approach along with classical biological techniques. For example, we could take worms, introduce human p-tau into them, expose them to DDT, and just kind of go back and do this whole analysis again on them. And show, we actually found changes in small molecules that corresponded to what we saw in the CSF of people with Alzheimer's in these worms. So we're not as different from worms as we like to think. There's a lot of things that are very well conserved in that. And I think this also can help drive other rodent studies as well. Quick little jump here over to Parkinson's disease for a second. This was a project we had, this was from the EPIC European Cancer Study. Hundreds of people developed Parkinson's disease. After that, about 8 years, on average was, so we had biological samples collected from them. Average age later, they had developed Parkinson's, and they had self-reports of coffee consumption. So they could estimate caffeine consumption. And I'm like, well, why don't we measure caffeine metabolites? And so my postdoc measured all these different caffeine metabolites, including the one we discovered the year earlier and put in the database that we wouldn't have known was there had we not put it in there. And what it shows is that people, like, we know that caffeine is protective in Parkinson's, but this shows that those blood samples from 25 years ago showed that the people that had higher levels of caffeine were less likely to have Parkinson's disease. So and it's not, I mean, not all exposures are like this, but many exposures are fairly stable. People drink coffee, their coffee habits are pretty similar. And so there's many things that you can get even with that snapshot in time to do that. Now in this one too, we've done the full untargeted GC and LC, and that data is also being analyzed. A little bit trickier because it was European, and so there's all these GDPR things, the work's being done in Europe for it. But it just shows that we can detect many of these different features and really do this sort of pathway analysis. So real quick, I'm going to talk about a project we have, should be starting in about 2 months. This is from the Advanced Research Project Agency, or ARPA-H. But what we were talking about was a lot of what Craig was even recommending was, if we want to look and see how the environment alters how drugs work, we have a lot of technology to do that. We just have to scale it up. And so what we proposed to do was, one, use the Odyssey EHR database. So this has electronic medical records for about 1 billion patients across the world, and the Mayo Clinic records, to look for suboptimal therapy, like which drugs are farther away from that clinical efficacy, like real world versus clinical trial. And then also to generate standards for about 1,000 FDA approved drugs and those environmental modifiers, like the grapefruit sort of molecules in that range. And then do a very deep dive with whole genome sequencing on over 10,000 people, where we have very comprehensive electronic medical records, so we can look at people who have different SNPs or different metabolites, and actually go beyond what I'm reporting on our LCGC part, also very deep analysis of PFAS and other emerging contaminants. And then use this to build pharmacokinetic models and network modeling for these chemical-drug interactions and drug-drug interactions, and develop CLIO-approved tests, so these things actually get into the clinic. And these would be targeted tests for that. And ultimately, just kind of create this automated pharmacoexposomics pipeline, the idea that we have automated sample processing, bioinformatic analysis, to identify essentially almost all the major drugs that people take. And then the goal here is to get this to a biobank scale, like, for example, like in All of Us, and think about strategies for commercialization on that. But this is a collaboration of multiple laboratories and a lot of different efforts to put all that together. But I think this sort of large-scale project is what's going to help get the exposomics forward to do it at that scale. And with that, I just want to acknowledge all the people that helped me do this, make a plug for our Exposome Boot Camp coming up in July in person in New York City. We have our new Center for Innovative Exposomics at Columbia that's working to build all these things out on what we're doing there at Columbia. And again, my plug here again for Exposome is that having high quality work in a journal dedicated to the field is critical for demonstrating to this scientific community what the exposome is. And so I encourage everyone to submit to Exposome. We'd be happy to see things from this conference. So with that, I'll be happy to take any questions.
Thank you, Gary.